Okay, very good morning all. So today I'm going to introduce about the mobile genetic elements which comes under the course of genetics and genetic engineering. So myself Dr. Rashmi from the Department of Biochemistry from JSS College. So what are these transposable elements? So these are the mobile DNA sequences which are found in the genome of many of the organisms and they are found in an abundant nature means they make up at least 45% of the human genome. Okay, so most transposable elements are able to insert at many different locations and they are relying on the mechanisms that are distinct from homologous recombination. Due to these characteristic features of these transposable element, they often cause mutation either by inserting into another gene and disrupting it or by promoting DNA rearrangement such as chromosome deletions, duplications and inversions. So these deletions, duplications or inversion due to the result of the presence of transposable elements on the genomic sequence. It is also having a varieties of names such as transposons, transposable genetic elements, movable genes, controlling elements, or they also called as jumping genes because they are moving from one sequence into another sequence within the genome. So in this picture, you can see the Barbara McClintock. She is a scientist who invented these transposable elements and she awarded a Nobel Prize for this invention. She observed a variegated kernels that are found in the corn. Okay, so kernels are the fruits that are present in the corn. So variegation means what? There is a multicolored formation in the seeds of the corn. So this multicolor formation on the seeds is due to the presence of transposable elements in the genome. So what are the general characteristics of these transposable elements? So many different types of transposable elements exist in the nature. Some have a multiple structures that encloses only those sequences which are necessary for their own transposition. That is, the help in the movement of the sequence within the genome itself. The another type is they have a complex structure and those structures encode a various number of functions, that is proteins, and they are not directly related to the transposition of the sequences. For example, the short flanking direct repeats. It is a sequence of about 3 to 1 base pairs long which are present on both the sides of the most transposable element. So, these sequences are the repeats that are found in the transposable element within the genome. So, in the course of transposition, these flanking repeats are created when their staggered cuts are made within the target DNA. This we can understand in the diagram that is 11.111 how the flanking direct repeats are generated when a transposable element which is inserted into the DNA. So you can see in the it is a double stranded DNA structure which is having a nucleotide sequence. The first we are going to select a restriction endonucleases enzyme which will cleave the specific segment of the DNA. Okay, so after cleaving, we are going to generate a staggered cuts. So the staggered cuts are made in the target DNA. The gaps in the staggered cut are filled by the transposable element in the second step. So a transposable element inserted itself into the DNA sequence. After the insertion of these transposable element within the DNA sequences, there is a gap which is formed. Okay, so the staggered cuts leaves a short single-stranded pieces of DNA, and these gaps are filled upon by DNA polymerases enzyme upon DNA replication. Okay, so these DNA polymerases introduces the sequences and this gap will be filled during the replication process. So here you can see in the double stranded DNA sequences, the gaps are filled by the sequences which are represented in pink color. So that is the flanking direct repeats. So replication of this single stranded DNA creates the flanking direct sequences in the DNA sequences. At the end of many transposable elements, as I already said, they are having terminal inverted repeats. What do you mean by this terminal inverted repeats? It is a sequence of about 9 to 40 base pairs in the length and that are inverted complements to one another. For example, in this, you can see one DNA strand which is running from 5' to 3' direction and another from 3' to 5' direction. So the nucleotide sequence is totally inverted from 5' to 3'. For example, AC, AG, TT, CAG from the 5' of a one strand is exactly inverted. So in the next strand, it is running from again 5' that is AC, AG, TT, A, CAG. These inverted repeats are coated on both the sides of a transposable elements, so which results in the formation of a terminal inverted genes that you can see next on the either side of a transposable element. Next, the figure 11.12 summarizes the general characteristics of a transposable element. As I already said, the transposable element is the sequence. Next to that, we can find a terminal inverted repeats. So, whatever the inverted repeats we have seen in the last picture, so that is represented in the green color. Okay, so next to that sequence, we are having a flanking direct repeats. Okay, so many transposable elements have these common characteristics. This helps in the movement of these sequences from one location in the genome to another location. Okay, hence they are called as jumping genes. Okay, so all the types of transpositions have several features in common. That is, they are having staggered breaks and they are made in the target DNA, and the transposable element is joined to a single stranded ends of the target DNA, and the DNA is replicated at the single stranded gaps and that will be filled by the enzyme DNA polymerases. So, some of these transposable elements transpose as DNA and are referred as DNA transposons. So, not only DNA transposons are present, we can also see the presence of RNA transposons in the cells. Okay, so in this case, the RNA is transcribed from the transposable element that is DNA and it is then copied back into the DNA by using a special enzyme called reverse transcriptase. So both DNA as well as RNA transposes we can see in many eukaryotic as well as prokaryotic microorganisms. Among DNA transposons, the transposition may be replicative or non-replicative. So what do you mean by replicative transposition? It is also called as copy and paste transposition. Means what? A new copy of a transposable element is introduced at the new site while the old copy remains behind at the original site. So let's understand this concept with this diagram. Okay, so here you can see there is a two DNA strands molecules that are represented as A and B. The A molecule is containing a transposable element. So before the transposition takes place, a single copy of the transposable element is found on only one molecule, that is A. Then in the next step, A and B is getting co-integrated. Means the two DNA molecules are joined and the transposable element is allowed to replicate. Okay, this producing a co-integrated molecule. In the third step, there is a crossing over at the region within the transposable element. So in the final, you can see both A and B DNA molecules are containing a transposable element. So this type of formation of transposable element in both the molecules is termed as replicative transposition. That is copy and paste. Why it is copy and paste? In the A it is present, the sequence, and it is copied, and again it is pasted in the B molecule. So it comes under replicative transposition. Next, what do you mean by non-replicative transposition? So the transposable elements move from one site to another site. It is example for cut and paste, okay, within the genomic sequences. So the non-replicative transposition requires only that the transposable element and the target DNA be cleaved and joined together. So cleavage requires an enzyme, that is transposase, okay, which are produced by the transposable element. Then the joining of these transposable element and the target DNA is probably carried out by a normal replication and repair enzymes. So this is an example. Here I have taken retrotransposons, which transpose through RNA intermediate. So in the figure, you can see there is a double-stranded DNA structure, okay, which containing a retrotransposon, which is represented in an yellow color sequence segment, and it is also flanked with flanking direct repeats on both the sides. So this transposons undergoing transcription, which produces an RNA molecule. Okay, so the retrotransposons sequence is transcribed into RNA. Then revert transcription, okay, it takes place on the RNA molecule and it will produce a double-stranded DNA molecule. Next, when you take the DNA sequence, so here restriction endonucleases are used. It is going to cleave the DNA sequence at specific site so that the staggered cuts are made in the target DNA. In this gap, these double-stranded DNA molecules are getting inserted. So here you can see in the double-stranded structure, the old copy of retrotransposons are also present, and here you can see this is a double-stranded DNA, that is a new copy of retrotransposons are inserted in the double-stranded DNA molecule. So copy and paste. So these are the retrotransposons which transpose through an RNA intermediates. So with this information, today I am going to conclude my class. Thank you.